Awesome. Thanks so much for having me. It's going to be super fun. I'm still cool aside that we didn't get to do this in person, but yeah, so today I'm going to talk about this idea of basically variable selection, where we map three-dimensional shapes and understand three-dimensional variation and how that can be used to explain genotypic or phenotypic variation. Okay. And so for those who kind of don't know a little bit of what I would do, a recurring research theme for my group is I like to think about taking modern computational approaches and develop theory that enable their interpretations to be related back to like classic genomic principles. And so how I like describe this is like, I'm not a statistician that builds really fancy hammers and then just like looks for nails to hit. I actually really care about the specific nail that is the biological problem, and I build a specific hammer for that particular problem. Okay. Now, a cross-cutting theme here is this idea of dissecting or understanding phenotypic variation. And when I say phenotypes, I can mean anything from, in humans, a lot of times I do genome-wide association studies where we study things like height, body mass index, type 1 diabetes, and we try to understand how the genetic architecture of these traits differ across different groups of people. Okay, so you can think about your phenotypic variance as being like an entire pie, right? You could break that pie into different components. Some of that might be an additive component, which is like the effect of gene A plus the effect of gene B. Some of that might be a pairwise interaction component. So you can think about the effect of gene A times the effect of gene B. You have third order interactions. You also have this nice little environmental component here where you think about how your environment plays a role or interacts with your genetics in order to give rise to some trait, right? And then you think about how that breakdown differs across different groups of individuals, right? And so here on the left-hand side, I like to show this plot where these are 131 different phenotypes from mice, broken down into different categories. What you could do is you can go in and you can kind of dissect whether some of these pieces from these, which effect is driving the variation the most across these different traits, right? And you can kind of see at the top, we have additive effects. Not only do you see that additivity very rarely kind of plays a role for some of these traits as the dominating factor that's driving these variation, but you can see in some of these traits, you have pairwise and these nonlinear effects also playing a role. So I'll talk about why that motivates my group at least to think about nonlinear models and machine learning as a way to kind of understand these kind of pies, right? This entire phenotypic variance. Now, what we're going to talk about today is not continuous traits, but modeling variation across shapes. So instead of things like height and BMI, I have maybe a collection of bird beaks, or I have a collection of heel bones from primates. We're going to spend a lot of time in this primate space as we kind of talk about stuff. Okay. So the presentation outline for me is going to be broken down into two pieces. Okay. The first piece is going to be this idea of what have we done in statistics where we work with shapes. Okay. So I'll talk about the history of shapes. Topological summary statistics, but this idea that we use TVA to enumerate or quantify shape variation. I'll talk about an application in radiomics, clinical imaging, and then I'll talk about this idea of SINATRA. And SINATRA is this pipeline that we created to do basically variable 3D selection, variable selection with 3D shapes. Okay, and so I'll go over the algorithm overview of the pipeline. I'll talk about how we do variable selection on each of these measures, and then how we kind of reconstruct these to kind of actually visualize enrichment on 3D surfaces. And then I'll show you some simulations that I think are pretty cool, and then some real data analyses. And I'll go over like what we're kind of doing now in this space. So previous work with shape statistics is actually quite interesting. And so what people used to classically do is you would take these three-dimensional objects, these three-dimensional surfaces, and you would use what we call landmarks, which are effectively placing, you would have an expert come in, look at a shape, place these landmarks on the shape, and those landmarks would serve as like a lower dimensional representation or summary of the variation that you're seeing for that particular object, okay? So instead of having this high dimensional skull, you have these landmarks, place things, and you can kind of compare across shapes as if something's different, I can have like this correspondence between landmarks, right? Now, obviously, the issue with this is if I have a radiologist or have an expert that comes in, I give an expert a shape, he or she might place markers on that shape for this particular object. Then if I give them another shape, they might place another set of markers that don't have a nice correspondence, like there could be variability there. And the classic examples, if I give an expert one shape, they place the markers on the shape, I take that object away and give them back that same object. They obviously could put markers back, but they probably won't be exactly the same. So there's high variability when you think about how experts do, but this is how we used to represent shapes in this space. Now what we do, which is often used for biological morphometrics, is we have these semi-automatically defined landmarks based on, which allows to bypass this variability that's caused by user specification, right? So what I do is I have these nice correspondences. These are teeth from primates. On the left-hand side, I believe you have a carnivore. On the right-hand side, you have a herbivore. What someone would do is they would come in, they would place landmarks over each of these shapes, and then you can define like a distance function, like a Procrustes distance or something, that then tells you how close in proximity or similarity this tooth is from this tooth, right? And you can do that over many shapes. Now, the issue with landmarks and what I want to do is, we don't want to just do these point-by-point comparisons. I want to study things across groups of shapes. So if I have many teeth that are observations from carnivores and many that are from herbivores, and then I have some phenotypic output, I want to be able to do variable selection on these teeth to identify, okay, what are the pieces of these teeth that are actually driving the variation between these two groups, right? And I want to do this over collections of shapes. And another thing that we want to do is I want to be able to also do these comparisons where I don't have explicit correspondence maps or these diffeomorphisms between shapes, right? So let's say I don't necessarily have a nice landmarking system for certain objects. Brain tumors is a great place that I'll kind of motivate this from, where there's not a one-to-one correspondence. How do I still do these kind of variable selection methods? So landmarks are great, but not necessarily great for what I want to do, per se. Now, what my group takes advantage of is a more a newer representation of shapes, and we use images or data that can be represented as meshes. And you're going to hear me say this a lot, which is effectively just collections of vertices, edges, and faces. Okay, so on the bottom here, you have a brain tumor that's been basically triangulated over. We create this mesh representation. And then what we're going to do with our topological data analysis, we're going to treat this mesh representation as a simplicial complex, and then we'll be able to derive things like Euler characteristics, persistence diagrams, these things over these meshes. Okay, so you're going to see me say these like vertices, faces, and edges statement quite a bit. Okay. So what's the main objective of like what I'm trying to do for statistics and modeling? Well, I want to be able to represent three-dimensional objects in a summary statistic or a form that can be used in a wide range of regression machine learning techniques, right? So I want to take shapes and quantify them in some summary statistic that I can use in both generalized linear models to neural networks to any kind of regression or supervised learning type of framework. And I want my desired properties. So when I go from shape to summary stat, in summary statistic space, I want to be able to define things like distances, define probabilities. But more importantly, I want the function that takes me from shape to summary statistic to be invertible. Because the goal of this whole thing is if I have a collection of objects, I want to be able to do variable selection in summary statistic space and then map back onto the actual shape and visualize those physical enrichments, right? So I need that invertibility property. Our topological summary statistics can be, you can choose many of them, right? And there are many invariants in the literature. We're going to focus on the bottom two, okay? So we're going to focus on the persistent homology transform and the Euler characteristic transform, okay? And I'll go over what each of these things are, right? I like to show this Picasso painting because it kind of shows like what we're trying to do, right? Which I'm sure many people have seen, right? We're going to work in this idea of this like lower dimensional type space, and we're going to try to understand what's happening in higher dimensions from this lower dimensional representation. Okay, so let's go to this idea of persistent homology, which I'm sure this is not new for a lot of people, but just so I can nail this idea home of why we use this mesh representation, because it very much happens, right? Persistent homology, we have this idea of a simplicial complex. Again, this idea of vertices, faces, and edges. And we're going to study this idea of homology groups, right? And so for those who, without going too far deep in the weeds of this whole thing, persistent homology tracks the evolution of homology via collection of these simplicial complexes, right? So I start with some data point cloud, some shape, I have some filtration operator, and I'm going to track how long certain features persist, okay? Now I'm going to get into why this is important in the context of our data in a second. But there are two ways to kind of track these features and look at this representation of this variation. The most classic is what we use as barcodes, right? And so you can track how different homology groups, connected components, circles, or voids track over this filtration. And you can do this by, again, I have these data point clouds. I grow these balls around these point clouds, and I could track how long certain things become connected, how long a certain hole persists, how long a void persists, and then I can compare that across shapes to see the differences of the variation across them, okay? Now, an alternative way of doing this is to use what we call a persistence diagram, okay? So the same idea of this idea of looking at a barcode, but now I'm going to just study the birth and the death time for each feature, okay? So here I have a two-dimensional representation. I'm going to do a filtration up, and I'm going to track when certain features are born and when certain features die, okay? And then I'll study how long certain things persisted, okay? So what we're going to do is I'm going to filter up this way. On the right-hand side, you're going to see my birth and death time pairs on the X and Y axes, respectively. So I'm going to filter up. I'm going to see my first feature. I'm going to filter up again. I see my second feature. So my first feature has been born. My second feature has been born at this part of the filtration. I filter up. I see another feature. Now that feature is going to die quite quickly. When I say die, what it means is it's going to become connected to my very first feature. Okay. So when I filter up a little bit more, that becomes connected. So that feature died quite quickly. Filter up. I see another feature. I see another feature. Now the feature just before that is going to die, right? Because it's going to become connected to my second feature. Filter up again. That one dies. I have one big connected. Okay. Now there are many, as I'm sure some people are kind of aware, there are many debates on what are we looking for here. But the natural convention for a lot of people is that things along the top, along the diagonal, things that are born and die quite quickly are topological noise, and things off the diagonal are topological signal. There's a debate based on that, but the idea here is that you can take these diagrams and compare across them to figure out different differences among these like topological features. Okay, so you might be asking like, okay, how can I actually use this in practice? So here's an example on 2D maze roots. What's really interesting about corn actually is you can study the topology of roots across crosses of yield across corn, and that's actually a really good predictor of how much yield that corn is going to actually produce. And so what you could do is you can take each of these roots. We create these persistent diagrams across them. I know which of these are crosses, so which how I like basically bred my different lines to create these different species of corn. And then I can compare across and try to figure out, okay, which species is going to produce the most yield, and then I can then backtrack and figure out which ones are the easiest to make, and so on and so forth, right? And so we could take these diagrams and find a distance function among them, and then figure out similarities across these crosses. Right. Actually,
Lauren, may I ask just a question about this slide? What is the filtration here? Am I like where? How should I? What direction am I reading?
Right. So we'll get into this a little bit. This is a 2D example. This probably should be 3D, which I'm going to talk about right now. Ideally, what you would want to do, and this is the Symbology transforms I want to talk about, is you would filter in one direction, you would rotate that object, and you would filter over like many directions over the unit sphere. That's ideally what you would do. And that's the persistent Symbology transform. And so you would look at these roots from like many different angles. That's a great question because that's my next slide. Is doing this for 3D shapes. Okay, so here we have a heel bone. Now the homology transform is very similar to just the first diagram in the 2D example, except now we want to do this over many directions to get an idea of variation of topology over many different faces for this thing. Okay, so how the Symbology transform works is I'm going to pick a direction for my shape. This is a heel bone from a primate. I'm going to define a filtration function or a height function. I'm going to sweep over this object. I'm going to collect my persistence diagram. I save it for that direction. I'm going to rotate the object. I'm going to define, I still have my height function. I'll sweep across that, and I'll do this over many directions of the unit sphere. So what you have for 3D shapes are collections of persistence diagrams, okay? And how you compare across shapes is you compare across these different persistence diagrams over these different collections, okay? So an example of this is, let's say I had heel bones from many different primates. So here we have 67 different genera. I do these homology transforms where I do these sweeps over many directions for each heel bone. So I have these collections of diagrams. I define a distance function to compare how similar they are. I plot these differences on some two-dimensional plane, and then we can like overlay to see that phylogenetically, heel bones are similar according to like, phylogenetics and selection, effectively, how similar certain primates are to another in terms of their species. Okay.
Lorin, can I ask a question? Sorry, this is Alex. Can you go back one slide? Just I want to make sure I understand.
Yep.
Here, this is a 3D shape.
Yep.
So you choose a direction, meaning a vector.
You choose a height.
Like a normal vector to the plane, or are you choosing an actual? So I'm confused whether you're scanning like a plane through it, or
right. So you're scanning a plane through like the point line. So in practice, what you would have done, you would have created like you would have represented this as like a mesh object. You would have swept through this you would swept through like as a plane all the way through the shape. It's going to be discretized because it's not going to be continuous, and so there are many things about everything I'm going to show you today, which are different parameters you want to think about in practice. But effectively what you do is you sweep through, you track these diagrams as you like discretize over.
So the diagrams are now two-dimensional.
Right. But you have it for that given direction.
Okay.
Right. Yeah, great question. Yeah, there are a few nuances I definitely want to think about and pose here. But I'll wait till I get a little further down to talk about this. So there are two things that are not great about the precision homology transform for my purposes. The first is, the precision homology transform doesn't produce summary statistics that have an inner product structure that I'm like we're used to, right? So in basic regression models, right, you have some inner product structure that's defined as some Hilbert space. The precision homology transform is going to give me these collections of birth and death time pairs. And a classic example of this is, what's the interpretation of an effect size for some like birth and death time type of ordered pair? It's hard to think about that geometry of that space. And so what we do instead of this is homology transform is we work with the Euler characteristic. And the Euler characteristic is this very simple equation here, a vertices minus an edge plus face. The same idea, except now I have a single number instead of like these birth and death time order pairs. Okay. Now the same idea is going to happen. I'll show this again in 2D, and then we can talk about what happens in 3D. I have a two-dimensional hand here. I'm going to create this, what we're going to call an Euler characteristic curve. How it's going to work is I'm going to have a filtration. Going back to the same idea, what I would do in practice is we would take this hand, we would represent it as a mesh, right? So like a triangulation over this object. I would do these filtration steps over that. And what we're going to do at each step in the filtration is count the Euler characteristic up to that filtration point, move up in the filtration, count the Euler characteristic, and then that's going to give me this nice curve. Okay. And so we would have a filtration, count up to that point in the filtration or that sublevel set. That's going to be our Euler characteristic. We move a little further, and I continue to count until I have my entire curve. Okay. Now what we can do in practice is if I have a three-dimensional object, I would have my shape, I would do that sweep, I have a curve, I would rotate that object, do another sweep, I would have another curve, and what I would do across these different directions is concatenate curves together, okay? So at the very end of a filtration for a data set, what I'm gonna have is a matrix that's like n by p, right? N being the number of observations, p being the number of features, where each feature here is just like a Euler characteristic at a given step of the filtration, but you have just a bunch of concatenated curves, right? The Euler characteristic is nice because it is also, it's easy to think about invertibility here. You're able to bookkeep, right? As long as I step through, I know in what direction each Euler characteristic number came from, right? And I know what step of the filtration came from. So I can always kind of go back and figure out, okay, this Euler characteristic came from this certain direction in this step in the filtration, right? And so we can think about how to bookkeep in order to think about invertibility here. What's also really nice about the Euler characteristic is now I have a collection of curves that represent these topological summaries. But the nice part is these curves have a nice simple inner product structure for which I can think about, right? I have one summary for every single topological summary statistic, one number, and I can think about what an effect size mean here. So I have this nice inner product structure that I'm used to in regular statistics in regular machine learning. And so I can also think about how to now open up this idea and use a full scope of parametric and non-parametric methods with this. Okay. So let
Lorin,
me kind of,
may I ask a question about the previous slide again?
Sure, sure.
When you do this for all of the examples in your data set, do you have to like pre-register your shapes so that they're all sort of approximately the same scale and like the orientations make sense together?
It's a great question. In practice, we do, but you don't have to. And I'll show you an example of you don't have to. You actually don't. You can run things on unaligned shapes and still get actually really quite nice summaries at the end. This is something that a reviewer pushed me on for the paper, so I'll show you in a little bit, which I was actually really thought they got a great example. So let me show you how the Euler characteristics work in practice. This is an example from radiomics. Effectively, what we have here is I have MRI scans from 40 patients for glioblastoma, so a type of brain cancer. This data comes from the Cancer Imaging Archive, and each patient actually has matched genomic and clinical data in the TCGA. So the TCIA and TCGA are work together in certain different data sources. So here's what our data looks like. We have an MRI scan on the left. What we do is we go in and we segment out the tumor itself, and so you have just the tumor on the right hand side. We have these segmentations for different slices for every individual. What we do is we go in and we run our topological sweeps, and so we get these nice curves, these Euler characteristic curves for every single patient. So our data looks like this. What we do with these curves is we're going to use like nonlinear regression with these models, right? So as soon as I have these curves, again, my group thinks about nonlinear models, going back to this idea of studying all types of variation. In this particular paper, we think about this from a GP perspective, right? Where we put a prior over this function space. You can think about K, the covariance function here, as summarizing the similarities of the topological profile for patient 1 versus patient 2, and so on and so forth, right? So you can think about any kind of covariance function here. We're thinking about it, let's just use a Gaussian for now. And so the idea of this project was, can we use these topological features as predictors of different survival outcomes? So if I had every patient and I had their survival outcomes, is the topology of the tumor a better predictor of these survival outcomes than things like gene expression, tumor morphometry, volume and geometrics, right? These measures that people typically take in the field when they're looking at imaging. And so I want to kind of focus a little bit on these two clinical outcomes. Here we look at disease-free survival, where disease-free survival is defined as the time from when a patient is diagnosed to the time that patient suffers relapse, so the time the cancer comes back, okay? Overall survival is defined as the time when the patient was diagnosed to the time when they actually passed away, okay? So what we do is we take each of these features as predictors in our model. We do these, we run these regressions, we perform these 80-20 in and out sample splits, and then we see which of these predictions lead to the lowest root mean squared error, right? So here are these results. So on the left-hand side, disease-free survival, right is overall survival. You have root mean squared error, lower number means better. This PR optimal is this idea of which percentage of the time was this particular feature the best out of the 100 runs, okay? Now, overall survival, I want to kind of draw attention to the right-hand side. I look at the right-hand side and I don't see a big difference. Effectively, these numbers are all the same. Overall survival is really murky in the sense that, you can be diagnosed and pass away, but a lot of things can happen in that time frame, right? Disease-free survival is much more well-defined, right? It's based almost primarily on the disease itself, okay? And so let me kind of tell you where I started going with this, which is like, wow, this is really cool from a prediction problem, but wouldn't it be really cool if we could do variable selection, right? And stick with me a little bit. So let me kind of give everyone like a small crash course in microbiology, something I'm like super passionate about. So you can think about our bodies as well-oiled machines, right? In our body in a normal state, in our cells get receive growth signals. The growth signals activate genes. We can think about a pathway of genes as like this linear thing, where the growth signals activate the expression of gene A or the signaling of gene A. Gene A talks to gene B, B talks to C, C talks to D. And in our regular healthy cells, things survive and proliferate and grow, right? So our cells grow and they move. If there's any kind of irregularities, cells have a system within them to tell themselves to apoptose and die, right? So basically kill yourself, right? In a cancerous system, in an oncogenic state, what happens is you get a mutation. So B is always on, which that means that this pathway of cell or genes is always constitutively active. Okay, so B is always on, C is always on, D is always on, D is always downstream telling the cell survive, proliferate, grow, no matter what. And then you get this idea of irregular growth in tumor, right? Now, what a lot of us have now are in a lot of different cancer subtypes, we have these agents that can come in and block the activation, these targeted therapies that come in and block the activation of certain genes or mechanisms in our cells, right? So basically, this agent comes in, B is now always off, because this agent is blocking its activation, which means C's off, which means D's off. The cell now is able to apoptose, and you look like you get someone who looks like they're healed, right? Now, the ZC survival studies this idea of relapse, right? Relapse happens when, even in the context of the drug, either this pathway turns on or another set of genes basically carries out the deed of this pathway. Regardless, downstream, survival and proliferation is always happening. And that's even in the presence of drug. And so you have this idea of therapeutic resistance, right? Now, what that last paper, that radiomics example kind of made me start thinking, well, if this is a nice predictor of disease-free survival, is there kind of a structural fingerprint of tumors that you can use or identify that are directly related or that are signatures of this type of underlying molecular activity? Effectively, as a scientific question, can you use 3D shape variation in medical imaging or oncology as a way to explain underlying biological phenomena, right? So this takes us past prediction, but now I want to use these Euler characteristics as a tool for variable selection, okay? And that's what brings us to this idea of SINATRA, okay? So SINATRA is the acronym stands for Sub-Image Analysis Uses Topological Summary Statistics. For myself, it has a little bit of a deeper meaning. My group does a lot of things. We do genetics, genomics, machine learning statistics, this weird TDA type stuff. And SINATRA is like the first project I think I've had in my group where we've taken these like weird disparate sounds and kind of had them in one cohesive kind of thing. And so I kind of like SINATRA as like this has this kind of double meaning for me. But SINATRA is just a pipeline of how to do this variable selection with 3D shapes, okay? So what you do is you start with meshes from two collections of, or two groups, data from species 1, or I have teeth from a carnivore, data from species 2, I have teeth from a herbivore. What we do is we transform these shapes into topological summary statistics. So we take these shapes, represent them as meshes, run these sweeps over them. I create these curves for every shape in my objects. So I have curves for species 1, I have curves for species 2. What we do with these curves is that we do variable selection on these curves, right? So we identify which pieces of these curves best explain the variation between my two groups. Once I've identified the significant pieces of these curves, right, the significant topological features, I then map back onto the actual shape the pieces of these curves that correspond to those topological summary statistics, and then you're able to visualize which structural features best describe the variation between a plant eater and a meat eater, right? And so SINATRA is a pipeline. So what I like to tell people who are in the math and applied math and statistics and machine learning cultures, I'm going to describe the 4 things that we do with SINATRA, but SINATRA is a pipeline. So if you hate any of the steps that I'm going to do, you can remove that step and then replace it with your favorite thing, right? But the 4 general things for SINATRA, there's 4 general steps, regardless of how you do them, is you want to take shapes that you're able to build summary statistics that represent or summarize their topology and geometry. You want to use a statistical model to be able to classify these shapes based on these summary stats. You want to derive an evidence of association metric. I'm going to, for each topological or geometric feature, and I'm going to show you what we do, but you can use p-values, Bayes factors, inclusion probabilities, whatever you love. And then where you can also, and then you want to have a way to project these features, the significant features, back onto the original shape so that you can actually visualize it on the actual mesh. Okay. So we already talked about the first step. Let's use the Euler characteristic. I'm going to stick in this Gaussian process space. So let's use the GP as what we're going to use to classify shapes. So now we need an association metric. Okay. Now, going back to my motivation for using nonlinear models, I love nonlinear models because, again, when I think about phenotypic variation, nonlinear methods are able to implicitly or explicitly encode these interaction terms that linear models are going to miss. All right. So if I have some type of variation of a trait, the linear model is going to miss that nonlinear component. Now, the interesting thing about linear models, though, is that we have nice rules for hypothesis testing and variable selection that we don't necessarily get for free for nonlinear methods, right? And so what I'd like to think about as a researcher is think about how to work in this nonlinear space and think about how to build theoretical techniques and tools that allow us to get interpretations like we're able to do in linear models, right? And so I go back to this idea of the kernel trick issue, right? Where I can do really nice linear models, and these betas are really important because they give us some notion of hypothesis testing effect sizes. But if I really want to be really good at prediction-based tasks or understand the total realm of variation, you really want to work in this n-dimensional function space, RKHS, where you can explicitly or implicitly have these like nice non-linearities. Now, if I work in that space, though, for me, as a computational biologist, or when I think about it, I really want to understand how my kernel machine, my GP, is upweighting or downweighting certain features in order to make these really nice predictions, right? I still want to be able to think about this in terms of like my actual input data. Well, the kernel trick issue is that there's no kind of invertibility back to your original input space, right? And so the issue with the kernel trick or things is like, once I make this transformation, I kind of lose the classic idea of variable selection, right? So I thought about this a lot in my PhD, and a little bit since then, is this idea of how to build effect size analogs for nonlinear methods. Okay, so what I mean by an effect size analog? Well, I teach Stat 101 and linear regression for PhD students. So let's think about this from the classic linear models perspective. Linear regression, I have some y equals my linear combination of my data plus some random noise. An effect size, right? An effect size estimate is me taking my data and projecting it onto the column, like my output y, and projecting onto the column space of x, right? And I can define this projection in any kind of way, but we can use least squares here, and just define this as like some generalized inverse, right, as my projection. Now, in a nonlinear model, I don't have this x beta, but here I have this like y equals some function plus some noise. The effect size analog is quite similar, except instead of projecting y into the column space of x, I can project my smooth nonlinear function f onto the column space of x. Okay. Now we can use any kind of projection we want here, but the idea is that let's just keep it simple. So we really have this juxtaposition between linear regression and nonlinear models. We could use a inverse, generalized inverse here as well. Now there's actually a lot of work in this space being done at places like UChicago and UT Austin, where people are thinking about how to specify this projection in different ways to pull different features out from whatever you learn from your nonlinear function. But here, let's just keep it very simple for the context of this talk. So, what can I do to get these nonlinear, these effect size analogs? Well, I went to Duke, as John mentioned, so I was just be Bayesian about it. Let's say that I have some y equals, I have some data, I put some priors on things. I could take a weight space view of my Gaussian process. I could fit an MCMC where I'm drawing new draws from my data f, from my function f, and every other hyperparameter in my model. But the nice thing is, like, once I define this deterministic step of whatever my projection function is, I can deterministically determine what a beta tilde might actually look like, right? And so I can get this like nice posterior predictive distribution and things like that, because every time I see a new f, I implicitly get a new draw for beta tilde. Okay. Now, these beta tildes are great. They're nice predictive weights, but the issue still remains that I don't have what I naturally get for free in linear regression, right? I don't have a p-value. I don't have an inclusion probability. I don't have a Bayes factor. So what do? Right? I just have these collections of weights. Well, my group also recently came up with this idea of doing variable selection by thinking about centrality amongst your variables, right? So let me kind of walk you through how this works. Like, let's say I have let's say my set of features in my data set, I have a sports team, right? With a bunch of players. Now, collectively, the sports team has a level of information, like as a network, right? This sports team has some built-in information. Now, what I can do to figure out how important each player is on the sports team is I could take each of them and put them on injury reserve, right? I could put them out, right? So let's say I put number 30 in the corner and I put him on injury reserve. Without number 30 in the corner they're playing, I might have a little bit of shift or loss of information about my network, but maybe not as much, right? The team's identity maybe as a whole stays intact. Now, let's say I take another random person, anybody, and that person goes and decides that they're not gonna play basketball anymore. They're gonna play baseball or shoot Space Jam for a little bit or something like that, right? Without this individual not on the team, I might lose a lot more information about the identity of my network, okay? Now, we can think about how to quantify this in statistics by using like KL divergences, right? The distance between how much I have when the full information is available to me versus this conditional setting where I've put my player as not being around, right? So the way to summarize this is studying the KL divergence between those two distributions, right? The KL divergence between the conditional distribution with the effect of that variable of interest being set to 0 and the marginal distribution with me having marginalized over that variable's information, right? Here, the KL divergence being 0 can be interpreted as that variant, J, is not a key explanatory variable relative to the others, right? In other words, the KL divergence is only going to be 0 if and only if those two distributions are the same, right? So what we use in our thing is this idea of rate, or the relative centrality idea, which is one way to naturally determine significance is to scale these KL divergences so that they're on a scale from 0 to 1. What this does is actually gives us a really nice null distribution. So the null distribution of rate, or the null hypothesis of rate, is that every variable in my data set is contributing equally, right? This idea of 1 over p, p being the number of features in my data set. Going back to my sports example, the null hypothesis of rate is that I have a bunch of bench players on my team already, right? The alternative is that some variables are explaining much more variation than others, okay? So let me show you how this works really quickly in terms of quick simulation. You can ignore most of this. Here, what I'm going to have is I'm going to have 2,000 samples, 25 markers. My markers are going to be, I'm going to have 3 of those markers be causal. So those are going to be my Jordan, Pippen, and Rodman. And then I'm going to run a GP, derive the effect size analog, and get rate from them, okay? So I run my model, and this is what we get. These are my Jordan, Pippen, and Rodman metrics here. You can see that them at the end, they're the ones that get selected, and everyone else is not selected. On the y-axis, sorry, is this idea of the rate measure. Then that red line is that 1 over p line. This is that line where I have nothing but bench players on my team, right? Now, to kind of show you this idea of what does centrality mean, now let's say I start removing players from my data, okay? So let's say I have Jordan, like Jordan affected, let's say I take Jordan, he's on injury reserve, and I iteratively put every other player on injury reserve with him. So Jordan's sitting out always, and it's everyone else sitting out with him. What you can do is see that if I remove Jordan from the data set, Pippen and Rodman are still the most effective or most significant features in my data set. But the relative importance of the rest of the features, which are effectively just noise, went up. And they went up uniformly, right? Because they're just noise variables. It's like saying if I lost 40 points a game, those 40 points have to come from somewhere, everyone has to step up. But everyone else's role in terms of the variation of my output is equal. So they all rise equally, right? Now I can do this again by taking Pippen and Jordan out. I just see Rodman, and every other player is kind of hovering around this 1 over p line. I can remove Rodman. You can kind of see we have this nice uniform distribution over each of my features, right? And this is the idea of the centrality, right? If I don't have a causal feature seen in my data set, everyone else is contributing about equal to the output. Now I can run sanity checks and say, okay, what if I remove number 30? You can see everything's kind of similar. And another null hypothesis say, what if I simulate data under just noise? You can see everyone kind of hovers around this line. And so that's the idea of rate. So we use rate in our model to basically assess how each topological feature is important or not important. Okay. So the last part of the algorithm is to map selected features back onto the shape. So again, you can use any kind of variable selection method you want. But once you have significant features, you need to map them back onto the shape. And so what we do here is we take advantage of this idea that my previous PhD advisor and some of his colleagues came up with this idea that directions, if I do a sweep over a direction, directions that are near each other are going to share similar information, right? And so what we do here is we leverage that. And so we pick, what we do is we pick cones of directions for our sweeps. We try to identify the significant features that fall within that cone. And then we save those, and then we repeat this for many different cones around our objects. And then what we do is we take the union of all those and reconstruct our shape. So let me show you how this works visually. I have a shape. I'll pick some cone of directions that we filtered or swept through. I'll look at the intersection of these as where do my significant features lie for that shape. I'll save those. I'll pick another set of directions. I'll look at the intersection of those significant features. I'll save those, and I'll reconstruct my object. Okay. And so that's the very last step of the SINATRA algorithm. Now let me show you how this works in practice really quickly, since I know I'm quickly running out of time here. Simulations were not free here. So I actually took for granted how easy we have it in other areas of statistics. In genetics, if I want to do simulations, it's quite simple, right? Like I take real genotypes, I select what features I want to be significant. I give those non-zero effect sizes like you saw in the Chicago Bulls example. I create the synthetic output, and then I say, can you select those? It's hard to think about doing that with three-dimensional objects where you have a three-dimensional shape and you want to select a feature of that shape that controls the variation for some output. And then you do that for many different groups. So the simulation schemes we came up with in themselves, I feel like are novel for our purposes. And so I actually really appreciate reviewers helping us think about how to do this in a very systematic way. So the first here is a proof of concept simulation. Here we're going to simulate data based on 100 spheres. So I take 100 spheres. I'm going to create features on these spheres by creating shared cusps. So what I basically do is you see these blue regions on here. I take we select landmarks and we basically perturb them by elongating these like cusps, and we're going to do that across all 100 spheres. So those are going to be like our shared features across these groups of shapes. Then I'm going to have causal regions. So what I do is we select a certain neighborhood of vertices for one class group, and then we create these indentations. So those are the red regions, and then we do the same thing for the other set of 50 spheres, but in a different neighborhood, right? So I have these kind of causal and shared regions, and the causal regions, I'm going to ask SINATRA, can you identify the vertices in these causal regions versus anywhere else, right? And then we're going to do like these power analyses by seeing true positives versus false positives in terms of our ability to identify these. And so what you're seeing here is the results for this in 3 different scenarios. And this is a sensitivity analysis, see how well SINATRA is able to do here. We go from scenario 1, which is quite easy, to scenario 3, which is harder. And let me kind of explain to you how this works. In scenario 1, we have one causal region. That's quite easy for SINATRA because if you go back to my pi example, I have a few vertices only that are describing the variation across my shapes, right? So every single vertex here, like let's say we have 5 vertices, I'm only splitting this pi 5 ways, so I give each vertex quite a large effect size. So SINATRA has quite an easy task of identifying this. Now on the right-hand side, I have many more causal regions, right? So the heterogeneity is a lot higher. And I have way more vertices explaining the variation across my groups. So you can think about going back to my pi example, instead of 5 slices now, maybe I have 100 slices, right? So each slice is much tinier. And so every single causal vertex has a much smaller effect size. And so it's much harder for SINATRA to identify those smaller effects, right? And so you see the power kind of go down. We see the same phenomenon happening in many areas of statistics of variable selection. Genetics is a great one. Sparse traits, where we have SNPs with large effects, are much easier to identify than what we call polygenic, which are many SNPs, many mutations happening for a trait with very, very small effect. It's much harder to see that. And so you see a kind of nice analogy here with this other thing. We did some method comparisons between things. I won't spend too much time on this. This is a quite easy example. So we could think about even straw man approaches to what SINATRA is doing. You can see even the straw man approaches do quite well. And then other things like the limit shaped algorithms come from this group at Harvard, which was not used for this purpose, actually. This is not what the method was made for. We kind of like worked with them to figure out a nice apples to apples comparison, even though it's still not apples to apples, but that's kind of what we did here. But you can see, even in this very easy proof of concept example, there are many approaches that do well at this easy task. Going back to my null case, and I always want to mention this, SINATRA is only good for is not good for two tasks in the setting that like there's a null hypothesis where SINATRA doesn't work. The first null case is falls under the rate regime. Rate says every feature is contributing equally under the null, right? So that creates two shape scenarios. The first shape scenario is where I have two shapes that are completely identical, right? So basically everything's identical except a little bit of noise. What SINATRA is going to say is, well, every shape, nothing here is different, so everything's contributing equally, so you're just going to see a null case where SINATRA is not that useful, right? The second case is the case where you have two shapes that are completely different. Same thing, SINATRA is going to say, well, every feature here matters, but every feature here matters equally. So everything's the same. Like I don't really see I think this is another null scenario. So SINATRA is only useful in the middle of these two cases. It's not useful if you can't see the difference at all. Like if you basically have two effectively similar shapes, it's not useful. And if you can see the difference between the two shapes by eye, it's also not worth using SINATRA for, right? SINATRA only falls in the middle of these two groups. So let's go to one more simulation. This is based on characterization. So what we do here is a little bit harder of a task. We say, let's take a tooth, a real shape. Let's put landmarks on that shape. We'll put 3 landmarks or so for class A, and I'll put a different set of landmarks for class B. And then we'll basically make caricatures of these landmarks. We'll blow them up in different ways according to some equation, okay? And then I'll ask SINATRA, reconstruct that shape and identify the landmarks for class A and class B, okay? So that's what you're seeing on the far panel. Well, the same kind of thing that we saw in the other case holds. SINATRA is quite good at this when you have low peaks, so not that many peaks. And then if I increase the number of landmarks that I'm making caricatures of, it gets a little harder. You can see the power start to die off. Interestingly, though, the strawman approaches are not great for this data where you have much more curvature and not as stark of differences between the two shapes. So now there are a little bit more nuances between things. You can see the strawman approaches are basically just random noise. And the limit shapes algorithm is quite interesting. If you have diffeomorphisms, you have to give the limit shapes algorithm a contact map, basically connected points between this shape and this shape so that the limit shapes algorithm can run. Now, if I take that map and I misspecify it a little bit, you can see that blue line versus that peach line, it starts to die off. Like it doesn't do quite well if that map is misspecified. And I remember John's question, so I want to get to that idea of what happens when we don't have this alignment. So the last thing that we did is we ran this on real data. We try to pick examples where we had pseudo ground truth, where effectively I knew something of like, I can give people confidence that what we're finding is real. So what we picked here is we use the Tarsius as our pseudo ground truth primate, where the Tarsius have a cusp in their lower molar that is preserved because selection, it needs to eat and digest food a certain way that the other monkeys don't have. Okay, and our goal was to identify this cusp. And so what we do is we look at these different groups. The Tarsius cusp is at that bottom, as you can see highlighted in red. And what we did these pairwise comparisons of the Tarsius and the other primate classes. And then we said, how well can SINATRA identify that thing? So you can see here is that for different primates, like how much prominent it is as you kind of move up in phylogenetic relationships, than if you kind of compare teeth that are a little bit more similar to each other, and how well the SINATRA is able to identify these. Now, going to John's question, you can run SINATRA the hidden gem of this paper is that you can actually run SINATRA on unaligned things. And how this works actually is if I take Euler characteristics, so I have unaligned shapes like you have at the top, and I run these sweeps over them, I get a curve, but the curves are for unaligned shapes. What we show is in this paper now is that if you minimize the distance or you normalize across curves, across these topological summary statistics, and then you reconstruct the shapes, normalizing or centering and scaling and basically finding like nearest neighbors across the topological summary statistics allows you actually to implicitly align the mesh that you initially started with. So what we do to get to panel B here is we take each of these summary statistics, we do this normalization or this alignment in EC space, and then reconstruct the meshes, and you can see how aligned these meshes are. Now, it's not perfect, but the reviewers were great at this, is saying that, well, this John went out, you're running this data on aligned shapes. So like, even though you're not using correspondences, you're still kind of using correspondences. So they pushed us to say, can you do this without doing that at all? Run this on unaligned shapes to show that you can do this. So here's what all the teeth look like in our data using an alignment software first, right? And here's what the teeth look like where we align, we run things on unaligned shapes and then do our alignment in summary statistics space. It's not perfect, but you still kind of see kind of nice alignment from this thing. And so it was kind of cool. This is something we're actually kind of pushing on to see how much we can actually do with this kind of EC alignment type of thing with these summary stats. But the same kind of idea, you can then run your model on the EC aligned objects and get your results. So there's a lot of future ongoing work here. I want to kind of wrap up. I really want to move back to oncology at some point. The problem with the oncology example is that the brain tumors case is hard. Intra-class heterogeneity is high for tumors. For the primates, actually, it's a really easy example, right? Because phylogenetics, like selection, has defined that if your teeth don't look a certain way, you're not going to be able to digest your food and you're going to die. So, right, like selection has played a nice role in the morphology of the shapes we're looking at. In tumor space, things can grow, at least to my understanding, as however they want, right? So like you have high intraclass heterogeneity within given certain mutation classes that you don't have in the teeth example. So we're actively working to figure out how to move ourselves over. And the next thing I want to think about is, moving past just objects, but thinking more of like imaging. So like 3D microscopy or PET imaging. So things where I don't have meshes anymore, but now I have just like imaging like you see at the top here, right? So instead of treating things as meshes, we've been thinking about this idea of how to treat images as fields and kind of do the same kind of thing. So that's been fun. And as an intermediate thing, I've also been using we've been looking at structural biology. So this is SINATRA Pro, which Pro just stands for proteins, where we're basically doing the same kind of idea, but looking at structural biology and trying to define biophysical properties. And why proteins are amazing, actually, is because proteins can vary in flexibility. So proteins kind of sit in the middle between our very rigid examples and our very very flexible examples like you might see with brain tumors or something. And so it's been super fun playing around with this data as well. And so this should be out relatively soon. And so with that, I just kind of want to thank everybody. There's a lot of, this is a really huge effort from a ton of people in a ton of different places. And people have been nice enough to give us money for stuff. So I've always received that. Yeah, so here's some references and I'm happy to take questions.